So chapter 3 introduces basic principles of heredity. In this first recording, I will briefly introduce Gregor Mendel and his mono and dihybrid crosses. So what we will discuss here now is how genes are passed from generation to generation, from parents to children and through children to grandchildren, how factors such as dominance, segregation, or independent assortment affect this inheritance. Basic principles of heredity have been first formulated in 19th century by Gregor Mendel. He was an amazing person, and you can find more about his life and his work in YouTube video which I posted on Blackboard, which comes from series Greatest Genetics Discoveries. What I would like to point at here is that when Mendel did his studies, many other botanists also have been interested in the inheritance. However, they failed to define the major principle behind inheritance. So why Mendel succeeded where others failed? So there are two major points I would like to make. The choice of experimental subject. He studied easily differentiated characteristics in pea plant, which is reproducing fast, which has many progenies, and which existed in many varieties. However, the most importantly, every characteristic he picked up to study exists in two easily differentiated forms. Second thing, scientific methodology. Mendel adopted experimental approach and interpreted his results by using statistics, mathematics. He crossed hundreds of plants. He counted thousands of offsprings. He analyzed them, he statistically evaluated them, he recorded outcome and formulated hypotheses and actually verified those hypotheses. At the time when Mendel performed his experiments, many scientists believed in blending theory, which stated that if you cross two individuals with two different traits, offsprings will exhibit phenotypes somewhere in between, so these two traits would blend together. So first question Gregor Mendel asked was, is this true? So he asked, if he crossed peas with two different traits, let's say round and wrinkle seed, will the offspring exhibit intermediate trait, blended trait, or will they exhibit trait of one parent or another? He carefully prepared for these experiments and assured that only plants, pure plants, were cross-fertilized by doing it manually, and followed the offspring in F1 generation. So what he observed, after crossing homozygous round seed, which was homozygous dominant, with homozygous wrinkle seed, homozygous recessive, he obtained F1 generation, which was all identical. All seeds were round, resembling one of the parent. So there was no intermediate blending phenotype. So it seemed like the wrinkle seed, the alleles which encodes wrinkle seed, disappeared. So what he did afterwards, he continued his crosses and basically he left self-fertilized F1 generation, and in F2 generation, indeed, he observed reappearance of wrinkled seed. So the allele for wrinkled seed did not disappear. It was just masked in F1 generation. Interestingly, as I mentioned before, he counted over 5,000 round seed and 1,800 wrinkled seed, which gave him ratio 3:1. And you can see how this ratio have been achieved. If you go to F1 generation, you have heterozygous if you cross heterozygous, each of them can form two gametes, one containing dominant and one containing recessive allele. Now, during fertilization, each of these allele can randomly fuse with allele from other parents, and there are four possible combinations which can be created during fertilization, which in one of the offspring will be homozygous dominant, one will be homozygous recessive, and two will be heterozygous. However, all offsprings who possess at least one dominant allele will manifest the same phenotype. So based on this experiment and many other with different traits he followed, he formulated principle of segregation and the concept of dominance. So what principle of segregation states? That each individual diploid organisms, each gene possesses two alleles for any particular trait, and these two alleles segregate during gamete formation, and one allele goes to one gamete and another to another gamete independently of each other. So these two alleles which form one gene, one trait, segregate during gamete formation. Based on disappearance and reappearance of wrinkle seed and many other traits, recessive trait, he formulates the concept of dominance, which stated that if two alleles are present, one allele masks appearance of second allele. You see, if you have F1 generation, basically the genotype is heterozygous, so this individual possesses dominant and recessive allele, but you can see only dominant phenotypes. So dominant allele masks appearance of recessive allele. So here you just have outlined in details principle of segregation and concept of dominance. So keep in mind, each gene, each trait is encoded by two alleles. Let's say round seed heterozygous has dominant and recessive allele, and these two alleles segregate during gamete formation, and each one goes to separate gamete. And this gamete, you see, it's breaking down from diploid number to haploid number to each allele. Next question is, how does law of segregation relates to behavior of chromosomes during meiosis when gametes are formed? Before we answer this question, I would like to bring to your attention one fact. When Mendel published his results, nobody in the scientific community really paid attention to it, and his work was forgotten. It was rediscovered again at the beginning of 20th century when several botanists become interested in heredity, perform genetic crosses, and obtain similar results as he did. In addition, in 1900, Walter Sutton formulated chromosome theory of heredity. In this theory, he stated that genes are located on chromosomes, and each homologous pair is formed by one maternal and one paternal chromosome. So this maternal and paternal chromosome segregate during gamete formation. So now, if you put together Mendel's law of segregation and Sutton's chromosome theory, you can very easily understand behavior of alleles during gamete formation. So let's follow this example from your book. Two alleles, let's say of genotype dominant R and recessive R, are located on homologous chromosome, which of course replicate during S phase of not meiosis. There is a mistake in this picture. They replicate during S phase of interphase. So Once the chromosomes are replicated and cell proceeds to meiosis, during anaphase 1, homologous chromosomes are separated. You remember, anaphase 1 actually separates homologs, so the number of chromosomes is brought down from diploid to haploid. So we start with diploid, and after anaphase 1 we have haploid number after anaphase 1. So if no crossing over appeared, if it didn't take place in prophase 1, after anaphase 1, each chromosome actually consists of identical chromatid. If crossing over took place during prophase 1, you can see here that chromatids are no more homologous. They are not identical, better said. So, during anaphase 2, two sister chromatids separate, and you form either identical gametes or gametes which are not identical. So you can follow alleles behavior through following behavior of chromosomes and chromatid during meiosis. So now we will continue and we will extend principle of segregation to more complex crosses that will include alleles of multiple loci. Mendel also performed dihybrid crosses and formulated based on his result principle of independent assortment. Before we continue, I just want to stress one thing. Dihybrid crosses refer to crosses where you follow two traits. Monohybrid crosses refer to crosses where you follow only one trait. So let's look at dihybrid cross. So when Mendel performed dihybrid crosses, he asked question whether allele of traits, two different traits, behave independently of each other. So we will not go to great details of his work. However, what I would like to outline is how you can answer this question. So let's follow this cross where you cross round yellow seeds with wrinkled green seeds, where round is encoded by gene on one chromosome and yellow is encoded by gene on another. So round, wrinkle, yellow, green. Now, if you cross heterozygous in both traits during F1 generation, and if you take into account behavior of chromosomes during meiosis 1 and meiosis 2, you will understand that each of these individuals can actually form 4 different gametes. And during fertilization, there is random fertilization of gamete from one individual by gamete of another. So how do you solve this cross? Once you know what kind of gametes individual in F1 generation form, you can plot them into Punnett square, where you put on one side all gametes formed by male and on another side you put all gametes formed by female. Now, if you know that fertilization is random, so any female gamete can actually fuse with any male gamete. So this cross gives you 16 different genotypes, which forms 4 different phenotypical categories. You will get 9 round yellow seed, which means that they have at least one dominant allele for shape and one dominant allele for color. 3 round greens, which means that they have one dominant allele for shape, and they are homozygous recessive for color. 3 wrinkled and yellow, which mean they are homozygous recessive for shape, and they have at least 1 dominant allele for color, and 1 wrinkle green, which means they are homozygous recessive for shape and also homozygous recessive for color. So what does this results show you? That allele encoding color separated independently of allele encoding shape. So even though the parents had different combination of allele of these 2 genes, this allele of these 2 genes can actually recombine independently of each other in F2 generation. So this data led Mendel to formulate his principle or law of independent assortment, which stated alleles at different loci separate independently of each other if those loci are located on different chromosomes. Okay, now we talk about segregation, law of segregation, and independent assortment. If you compare and contrast those 2, in both of them alleles segregate. So what is the difference? So law of segregation refers to segregation of allele of 1 gene, while principle or law of independent assortment refers to segregation of allele of 2 or more different genes. So you can manifest law of segregation by using monohybrid crosses. However, when you want to manifest or confirm principle of independent assortment, you have to use at least dihybrid crosses. Principle of independent assortment is a result of the behavior of chromosome during anaphase 1, and it results from the independent separation or segregation of homologous chromosomes. If you look at cells which contains 2 pairs of chromosomes, let's stick to the same traits, round and yellow, both heterozygous. You know that this chromosome replicate during S phase, and during metaphase, chromosome align randomly at the metaphase plate side by side, and you can have these combinations. Now, this random alignment during metaphase actually predetermined separation or segregation of homologous chromosome in anaphase 1. And in anaphase 1, here, each pair of these homologous chromosomes, which were aligned randomly in metaphase plate, separates independently. So basically, if you follow the genes located on those different pairs of chromosomes, let's say one chromosome and other chromosome, you will see that gene on different pairs of chromosome assort independently, and you will receive new different combinations of allele in gametes. Last thing I would like to discuss is how you can predict outcome of dihybrid crosses using branch diagram. Why branch diagram? It is much faster. Of course, you can always construct Punnett square, but Punnett square for dihybrid crosses can take time. Branch diagram is based on fact that when the genes of 2 loci separate independently, a dihybrid cross can be broken down into 2 monohybrid crosses. You calculate outcome of these monohybrid crosses, and afterwards you combine them. So let's do it. So how do we break down this dihybrid? So first you calculate outcome of monohybrid cross, when you cross 2 round seeds, which are both heterozygous, And you know that you will obtain 3/4 of round seed which have at least one dominant allele, and you obtain 1/4 of wrinkle seed. The same you perform for color. You cross yellow seeds, both heterozygotes, and you obtain 3/4 of yellow seeds, meaning they have at least one dominant allele, and 1/4 of recessive, homozygous recessive, which are green. So this was broken down dihybrid cross to monohybrid. Now you have to combine them and ask. What you ask here? You ask how many of round seed will be yellow, so it means how many seeds would be round and yellow, or how many round seed will be green, so they will be round and green. So you use multiplication rule of probability, which we will talk later on about. The same you do for wrinkle seed. So let's do it. So let's combine. So first you have 3/4 of seeds which are round and 1/4 which are wrinkled. Now this round seed can be either yellow, so they can be either yellow, or they can be green. Or the same apply to wrinkled seed. They can be either yellow or they can be green. You remember independent assortment. Each one can combine, each allele of each trait can combine independently with alleles of another trait. So now, if you want to calculate how many seeds would be round and yellow, you just multiply the amount of round and yellow seeds. So it would be 3 by 3. 3 multiplied by 3 is 9, and 4 multiplied by 4 is 16. So we will have 9/16 of round and yellow seed. Now the same you apply for any other combination. So we will have 3/16 of round and green seed. You will have 3/16 of wrinkle and yellow seed, and you will have 1/16 of wrinkled and green seed. So you see, using law of independent assortment combined with multiplication rule of probability give you the option to calculate or predict outcome of dihybrid crosses very fast and very by simple way. So in next recordings we will talk in more detail about the outcome of genetic crosses and how can be predicted.